All right, folks. Hopefully, y'all having a nice little Wednesday. I'm recording this on Tuesday, 4/13/21, but I'm going ahead and getting ahead. I'm going to go over a little bit of material with you guys, the latest information that's been posted on the class webpage. Let me show it with you here. It is Unit 5, Lesson 3 on DNA structure and replication. So let's go ahead and take a look at this. You can see DNA in the background. It's kind of got this neat little double-stranded structure. You may find out about that word or phrase later. DNA is considered to be a double-stranded molecule or a double helix. All right, so we do have to talk about its structure and then how it replicates itself. All right, here's some words or some vocabulary for this section that you need to be affiliated with, or you need to be, I guess you could say, comfortable with using. You've got deoxyribonucleic acid, which is actually DNA. Nucleic acid, which is just, nucleic acids. Polymers, nucleotides, double helix, which again we mentioned earlier was the shape of DNA. Nitrogen bases, and there are actually 4 of those. They are listed here: adenine, thymine, cytosine, and guanine. How DNA replicates, how its hydrogen bonds are related to that, and how you end up with parent or daughter strands from one parent strand. All right, so DNA structure, right? Let's take a look at this. DNA structure is a nucleic acid, right? That's one of the 4 biomolecules. If you remember correctly, they were carbohydrates, lipids, proteins, and nucleic acids. DNA specifically is called deoxyribonucleic acid, and the sugar in DNA is deoxyribose. It is double-stranded, and it's in the shape of a twisted ladder. You can kind of see that right there. It looks a little bit like a twisted ladder, right? If I was to draw it as best I could, I would, say maybe it looks like that, okay? Now, at the end of the day, this molecule is made up of a series of nucleotides. These nucleotides are the monomers, right? Or they are the building blocks of all DNA molecules. Now, let's see if I can go over and find. I'm doing something on my computer real quick. Sorry about that. This right here would be a nucleotide, right? And a nucleotide has 3 parts. It has the 5-sided sugar, it has a phosphate group, and then it has a nitrogen base. All right, and it's the same thing all the way down. This double-stranded DNA molecule is a bunch of, there's a nucleotide, there's a nucleotide, there's a nucleotide. All right, then you turn them the other way and you get the other side of the DNA molecule. All right, so literally, DNA is a series of nucleotides or a sequence of nucleotides. Now, the people who discovered the actual structure of DNA was 2 scientists called Francis Watson and Crick, I believe was their name. They're the people who actually discovered the shape of DNA and called it a double helix, or like that twisted ladder. All right. And just FYI, I do want y'all to know that if it was a ladder, that the sides of the ladder are actually the sugars and the phosphate groups. They're all the sugars and the phosphate groups. And then these steps in the ladders, these would be your nitrogen base pairs, right? And you'll find out what they are in just a second. Well, actually, here it is right here, your base pairs. For every adenine, there's a thymine. So for every A that's on one side, a T will be on the other side. And for every G, there's a C. For every G on one side, there's a C on the other side. Matter of fact, you can go back, see these base pairs here, CG, AT, CG, GC, AT, CG. So for every A on one side, there's a T on the other side, et cetera, et cetera. So these are your nitrogen-based pairs, and then these outsides would basically be your sugars and your phosphates. They call that the actual sides of the ladder, they call that the sugar phosphate backbone. So you might want to kind of remember that. It is called the sugar phosphate backbone. All right. Now, excuse me. Again, this is about the building block of the DNA. It is a nucleotide. You should remember I told you it had a 5-sided sugar. So all of these that I'm circling are 5-sided sugars. All right, and there's the 5-sided sugar. If this is DNA, it is deoxyribose. Right, remember all sugars end in OSE. There you go. If it's RNA, it would be ribose. Now you got the phosphate groups, which all these are in green. You see all these phosphate groups attached to these sugar molecules. Here is our phosphate group here. And then finally, you have your nitrogen base. Here are all of your nitrogen base pairs, C with G, G with C. And in this image, your nitrogen base is right here. And again, both of these, this is many nucleotides that you see on the left, and then this is just one single nucleotide that you see on the right. Now, in DNA, you will get this question, or on the EOC, you will get a question about things being complementary, right? So I want you to remember that. What complementary means is they'll give you one side of a DNA strand, and what they're expecting you to tell them is what is on the other side. Now, complementary strands are held together by chemical bonds that are in the middle between these nitrogen bases. And these bonds, I'll write with little yellow lines, those bonds are called hydrogen bonds, right? They're very weak, and there's a reason they're very weak. The reason they're very weak is it makes it easy for DNA to split apart when it gets ready to replicate, right? But again, these bonds actually connect nitrogen bases across the two strands. So if I had an A here, it would be connected with T through a hydrogen bond. G's would go with C's, C's would go with G's, A's would go with T's, T's would go with A's, A would go with T's, and so on and so on. But just make sure you know that between every nitrogen base pair, there is a bond connecting them, and they are called hydrogen bonds. They are very weak. It makes DNA replication easy. That's a good thing. A bad thing is due to the fact your DNA and genes and all of that stuff can break apart so easy, you can end up with a bunch of mutations due to being exposed to X-rays or UV rays, or you could have some genetic recombination occurring, right? So that's the bad side. So always remember, for every plus, there is always going to be a negative, right? So that's what complementary means. And you can see that obviously these two strands, the left side and the right side, are complementary. Now, excuse me, again, this is just another picture of the same thing. DNA has two sides. So say they gave you that side on a test. The other side would be what is complementary, right? So that's this complementary strand that's in red. And all of this should match up. Let's see, A would go with a T, that's right, TA, TA, CG, CG, GC. Another way to remember this is apples go in a tree, cars go in a garage. That's an easy way to remember A, T, C's, and G's, right? So if A is on one side, T is on the other. If G is on one side, C is on the other, all right? And that is what a complementary strand is. It's just simply what's on the other side. Now, for DNA replication, this is the final part of this section. DNA replication is important, right? And it occurs during interphase, both prior to mitosis and meiosis. Now, hopefully y'all remember part of the cell cycle. The longest part of the cell cycle is interphase. And there's an S phase and a G phase that's occurring during interphase. S stands for synthesis, which means to create or make new DNA. And then G stands for growth. So that's when the cell is growing, and that's when the cell's creating new DNA. So anyway, during interphase, DNA makes an exact copy of itself for the daughter cells. That should make sense because during interphase, you have to make a copy so that when it divides again, if it's mitosis, the daughter cells have the correct amount of DNA. So for that to happen, this original DNA has to unzip. And that means it breaks those hydrogen bonds. Then when it does, it goes into two parent halves, right? This red come from the parent, this red come from the parent. And now what will happen, which you see this in orange in these pictures, the free-floating nucleotides will come in and bond to the open strands. And then you end up with two identical copies from one parent cell. Right, and the funny thing about that is these free floating nucleotides that come in and attach here to the original parent strand, you get these from the food you eat, which that's kind of wild because if you've ever heard this before, you are what you eat. Because when you eat something, you eat its DNA, and then that DNA eventually incorporates itself into your DNA. So that's kind of wild to imagine that. Also, remember, this is just another way of looking at DNA replication. Remember, chromosomes are traditionally an I shape, and then after interphase, you end up with these things called replicated chromosomes. All right. So in DNA replication, there are steps involved in that. The first thing that has to happen is that parent DNA has to unwind or unzip. So that parent DNA kind of unwinds and unzips. Both parent strands will now serve as a template. So you got template A and template B. And free floating nucleotides from the foods that we've eaten will come in and attach. And then we'll end up with two identical daughter strands from one parent strand. And this method is said to be semi-conservative, right? It's said to be semi-conservative because one half of the parent strand, right, is retained. And in other words, what that means is you keep half of the original parent strand. The other half has been, I guess you could say, manufactured or created. All right. Anyway, that should cover us with Unit 5. If any of y'all have any questions, as always, you can send me a message at Canvas, and I'll be happy to help you.